Okay, today I get to speak to someone literally across the world from me, hailing from Perth in Australia. His name is Guy Ben-Ary, and one of my listeners reached out and said, hey, there's a bunch of cool stuff happening here in Perth. Take a look at this guy. I looked at it and I really couldn't believe what I was seeing. It was pretty amazing. Then I realized I'd heard about this before. I started digging in. I reached out, and he was very happy to come and have an interview. So I reached out, and here we go. So rather than explain it myself, I am going to turn this over to Guy. Hey, Guy, how's it going?
I'm very well, thank you.
Thank you so much for taking the time out to have this chat. I am so curious about the work that you're doing. For people who aren't familiar with it, why don't we start off by having you give a description of what your current work is?
Okay, first of all, thank you for your interest in inviting me to do this interview. I'm an artist and a researcher in a very special, I think, special lab in Perth called SymbioticA, the Center for Excellence in Biological Arts. Again, as I said, I see myself as a researcher and an artist, and working in the world of new media, where the technologies that I use are emerging technologies, and the materials that I use are biological materials, and the activities that I engage with are somewhere between growing and making. Just a couple of words about SymbioticA, because I think that it's very much relevant to my work. SymbioticA is an art and science, or a research lab into biological arts, that is located in Perth in the University of Western Australia. And SymbioticA is quite a unique lab in a sense that it allows artists to engage critically and practically in practice in biological technologies. We formalized our relationship with the university, and the lab lives and is located in the School of Anatomy and Human Biology in the science faculty of the university. So it's not located in an art school or art environment. It's more in kind of the science kind of bit of the university. But at the same time, it's run by artists. The director, Oren Katz, and co-founder of the place, Oren Katz, is a biological artist as well, and he created this or developed this institution or research lab, and he was very strict with the fact that it would be run by artists and host artists, but it would be located in a scientific environment. And that's quite an amazing space for people like myself, because there's a lot of knowledge, there's a lot of scientists, there's a lot of people that are engaging with biological technologies, with biology, and with kind of newly acquired knowledge that we are kind of learning and producing these days in kind of those environments. But at the same time, the work is artistic. It's not there to communicate science. It's a critical work that is developed in this place. We get a lot of scientists that are collaborating with us, and there's quite a lot of interesting projects that are coming out of this lab. So this is a little bit about SymbioticA, and it's interesting because SymbioticA was the first lab as such in the world, and at the same time, there's not a lot of places like that in the world today. And Perth is such a isolated, it's the most isolated capital in the world, they say, but it's still one of the kind of world capitals in biological arts, which is quite interesting because of SymbioticA.
Mm-hmm.
I've been SymbioticA since its inception in the early 2000s, and my work through there, I produced quite a lot of work in SymbioticA, but most of the work that I've done is kind of focused on neural network, on consciousness, questions about consciousness or intelligence, emergence, and I've been looking at neuroscience, the interface between neuroscience and art, and most of the work that I produce involves the growing neural networks, so basically external brains outside of the body. I mean, neurons are using tissue culture techniques. Tissue culture is a technology or technique that's been with us for more than 100 years. It tells us that we can actually grow cells outside of the body. Neurons, brain cells, can be grown outside of the body as well. What I was really interested in is growing them in quantities that they would start producing or creating neural networks, real biological neural networks, and growing them on interfaces that would allow me access to and from those neural networks to communicate with those external brains. I just want to say that one of the motivations for this work is an ethical one. The realizations that that the newly acquired knowledge allows us to grow entities outside of the body to create life in labs. And not only that we can create life, we can create entities that may have the potential to be conscious or intelligent in the future.
Right.
We're not there yet, of course, but when the manipulation of life, as we do it in labs all around the world, in every single lab in the world, when it takes place in an atmosphere of conflict and profit-driven competition, the long-term result of those manipulations, of those creations, might be disquieting, because the question is who does what and who can do what. And one of the roles I believe that art can play in this world is to suggest scenarios of worlds under construction, to subvert the technologies for the purpose of creating contestable objects, for instance. Art can make the emergence of those living entities evocative, and the multi-leveled explorations of those objects very important and very relevant. So this is kind of one of the basic ideas behind the work that is kind of underlined it's there accompanied with all the work that I produced previously. So as I said, I was very interested in creating those living entities, or entities that may have the potential of becoming intelligent later on, or in the future, not later on, and ask questions about those scenarios that I'm proposing. So the technologies that I use are basically, the hardware that I use are the neurons. Those are the cells that I'm growing in the lab. I mean, these days I'm working also with heart muscle cells, with cardiomyocytes, but I'll talk about that later. But really, the wetware is the main kind of hardware that I'm using and growing in the lab. The software that I use is a stem cell technology that is called pluripotent stem cell technology, or IPS, that allows us to reprogram the cells to become stem cells, and then you can differentiate them to any type of other cells in the body. So, this is what I do with my own cells. I grow my own cells, skin cells that I can take through biopsies, and then I reprogram those cells to become stem cells, and then I differentiate them to become neurons. So this is how I achieve or get my neurons,
Sure.
Rather than harvesting it from myself or from other animals or species.
Right, right.
And the sensors that I work with, the interfaces, is the main interface that I use is called the multi-electrode array. It's an electrophysiology system that is consisted of amplifiers, but mainly a specialized petri dish that has an array of electrodes fitted on its base. So there's a grid of 8x8 electrodes that I grow the neurons on top of those 64 electrodes, 8x8 electrodes. And when I plate the neurons and I start growing them, they form the neural network, and they start producing electric signals on top of those electrodes. And those electrodes, they are the read and write interface. I can listen to what the neurons are doing in 64 areas of the dish. The resolution is 64 in the dishes that I use.
Okay.
But what's really interesting as well is that I can talk back to the neurons. I can actually send information to the neurons through those electrodes as well in a resolution of 64 electrodes. So I have a read and write interface for the neural networks. The neural networks, when they're plated, they're starting to produce data to, the way neural networks work. And what we're trying to do is to stimulate those neurons and to kind of see if through those stimulations that we send to the neurons, we can change the functional plasticity of the neurons. We can get them to change what they do, to change their behavior. We can get them to learn or kind of show maybe any signs of emergence. Now, it's important to kind of mention that it's not a brain. I call it an external brain. It's not a brain, because the brain, how a brain consists of, you know, billions of neurons and trillions of synapses, and I'm growing 100,000 cells on this kind of interface. But however simple or symbolic the brain, quote unquote, of course, may be, they do produce quantities of data, and they do respond to stimulations, and they're subject to a lifespan. So it's very interesting to work with them. When I started to work with those kind of neural networks, I was really interested in what's happening there. What is the data that is produced within those kind of neurons? And this is how I got into kind of the idea of embodiment. What I thought about is to give those neurons or neural networks or external brains, as I refer to them, a body. And the embodiment was a strategy to highlight the liveliness of those microscopic neural networks and to manifest their, I don't know, their erratic existence through movement and through behavior, and to create a feedback loop. So throughout the last 20 years, I produced various kind of artistic projects where I gave those neural networks various types of bodies, artistic bodies, robotic bodies, and those robotic bodies interacted through this interface with those neural networks to create a feedback loop between the brain and the body. And, you know, I mean, the work is concerned with the directions that those current emergent biotechnologies would take us in the future. You know, I'm asking what are the responsibilities that we have to those liminal lives that we create, or what sort of ethical boundaries do we need to establish around those living entities? Because when artists using neurons have the potential to evoke responses in regards to the shifting perception surrounding the understanding of life, we're trying to bring possible scenarios to life. The neural networks confront the viewers both instinctively as well as intellectually, I think, by calling into question the liveliness of those entities. You know, that when I say liminal live, you know, when people or the viewers don't really know how to treat them. Are they alive? Are they dead? What are they human? They're non-human. They're animated. They're not animated. So through this kind of work, I get people to try and think about what those living entities are. So the first project in this line of work that I produced, we called Me Art. That was I worked on it in the early 2000s, where we we kind of thought the embodiment that we gave, or the body that we gave to those neural networks, was a robot that drew two-dimensional pictures or drawings.
Right.
And one of the reasons that we made it an artist was that, you know, working with the scientists, that in this same field, you know, the embodiment that they're kind of looking at, it was always utilitarian, you know. And we kind of try to think of what would be the least utilitarian body that we can give those neural networks. So we made it an artist, of course.
Right.
You know, the scientists
An
would
illustrator, right?
Yeah, an illustrator. So this is kind of how it started with this kind of project. But I guess that I would like to go into more detail describing the current project that I'm working on. Through the years, I produced a few other projects like Silent Barrage or In Potentia, various kind of types of embodiments. But with the current project called cellf, CELLF, this project is my self-portrait. So I applied to get funds to create a biological self-portrait. And what I've done was I went and I did a biopsy, and I harvested skin cells of mine. And I grew those skin cells in the lab, and then I reprogrammed them to become stem cells. And once they became stem cells, I differentiated them to neurons. So I, in effect, grew my own external brain outside of my body. And that was quite an interesting process. 2 years of, you know, looking at myself through the microscope, trying to figure out how to get myself to do what I want those new cells to become.
Okay.
And once I managed to kind of grow those neural networks of my own external brains on this interface, I was wondering, how would I portray myself? Where is this kind of biological self-portrait? And, you know, as I said, for 2 years, I looked at myself through the microscope, and the last thing that I wanted to do is to portray myself. I got a little bit sick and tired of myself, so I decided to portray one of my childhood dreams. And when I was 13 years old, the only thing that I wanted to become is David Bowie. I wanted to become a musician, and I'm not a musician. I'm not a musician at all. I can't play on any instrument, but I do have quite a distinct taste in music, and I have a lot of musician friends, etc. And I always kind of thought about how would it be to be a musician. So I decided to create my alter ego rock star, a rock star in a dish, and to embody my external brain, or those neural networks that I created for my own cells, with a sound producing body. So then I called a musician friend of mine, Darren Moore. He's a collaborator himself, and I told him about this project. I shared a house, an apartment with Darren in the early 2000s, and he's a drummer. He's a great drummer. He lives in Tokyo now, and he's very active on the experimental scene in Tokyo. And but when we lived together, I kind of kept on saying, "Hey, let's play together. I'll sing. I don't know how to do anything, but I'll sing." But it wasn't really serious. He always kind of dismissed me with, get your shit together. If you want to be a musician, be a musician. No shortcuts here.
Right.
And I called him and I said, "Hey, we are going to be on stage in 2-3 years from now. I have my ways." He started to dismiss this conversation again, or this idea again, until I told him what I'm planning to do, and then he got really excited. We decided to kind of collaborate on this project. Because I'm not a musician, I thought that I would need someone that knows what he's doing when we kind of looking at the sound aesthetic and kind of structure and improvisation and noise, etc. So, and he was the one that kind of proposed that I work with analog modular synthesizers. And the minute that he kind of proposed that, it kind of made sense to me, because in a really weird or oversimplistic way, analog modular synthesizers work in a very similar to those neural networks. There's a control voltage that is kind of passing through or flowing through components, whether those are the neurites or the modules of the synthesizer. It's like a CV,
right.
Control that is kind of flowing to produce data, whether it's kind of neural data or sound. They work in a very similar way. And when I hooked up with Andrew Fitch, the synth builder, the guy that is actually building the synth and again, self-collaborator. When I hooked with him, he kind of suggested, and I couldn't agree more, that we just keep the whole project analog.
Right.
Self doesn't have any computers, microcontrollers, programming, nothing. This is why the analog synthesizers fitted this project so well, because the signal flows from the neurons through this interface to the analog modular synthesizer with no intervention. It's like one network that is kind of flowing from the micro to the macro.
So the neural interface isn't computerized at all. It's literally just sensors and amplifiers?
Absolutely, yeah.
Unreal.
There's no computer. Because the thing is that, I have a rig in the lab that does it through, you digitize. Right. You get the neural network, you get the signals for the neurons through the A to D cards, and then you kind of see the data on the computer. But Andrew said, well, it doesn't make sense to digitize it and then to turn it into analog again.
Right.
Let's keep it all analog. And when I said, well, I would love to do that, but hey, I don't know how to. And he said, well, I'll build this rig for you. Mm-hmm. So Andrew actually built the equivalent of this rig through hardware. And it actually works better than the original one, because there's no conversion. The signal is very, there's integrity of signal.
Yeah, very direct,
so,
right.
Yeah. So with Self, what we're doing is not only that the neurons control, they control the analog modular synthesizers through this interface that Andrew developed. I didn't want to be a musical instrument. I wanted to be a rock star. Right. So we created this platform where we invite human musicians to play with Self, and the music that is produced by the human musicians is streamed to the neurons through this interface and modules that we developed, and the neurons respond to those stimulations by controlling the analog modular synthesizers. And then there's a feedback loop between the human musician and the non-human musicians, which is happening all the time. And they're basically jamming in a very similar way to the way musicians improvise in kind of a free jazz concert.
Right.
So it's basically like a post-human performance. Um, and I think this is kind of, in a nutshell, Self and my work.
Yeah, it's really quite amazing to kind of consider this as, I mean, when I wrap my head around it, it's kind of bizarre. Now, I have like 50 questions to ask about this particular project. So, first of all, you're working with Darren. He's literally playing. It stimulates the cells, which generates some voltage that plays back, and then he's responding. So it's literally a two-way communication between the drummer and the cells.
Yeah, definitely. And we also created these modules that we developed. Not only that it kind of stimulates the neurons, we created those gates that allow the human musician to that allows the signals from the human musicians to access to the neurons only through a certain range. So basically, the human musician, can also decide when to stimulate the neurons,
Oh,
which is kind of even closer to a setup where two musicians are playing together. If I'll play the solo, it's drum solo, and you'll play the bass, we would have kind of, we would look at each other and would have various kind of signals on when do I do what and when do you do
right.
your solo. And through this kind of free gate system that we developed, the human musician can actually signal the neurons by stimulating them in certain kind of times and certain kind of ways. I just want to kind of say that the project is not limited to Darren as a collaborator. We already had like 9 performances, and in each performance we played with a different human musician.
Okay. Going to say one of the things that, if you think about collaborating musicians, there's like these tacit communications, and also very subtle exchange of information. And it sounds like you actually have built a mechanism to do that. Did you say it's like through is it like different frequency ranges, or is it
Yeah,
kind of like spatial ranges?
different frequency range.
I see. Okay.
And again, I mean, it's you know, uh, let's be frank. I mean, it's quite symbolic,
Right.
but
Right.
it's still there, and the musicians can decide to use it or not
Now,
when they.
what are the kinds of responses that occur? I mean, so you say that this is roughly 100,000 cells that make up
Yeah,
this neural network.
yeah.
What are the kinds of things that, when you interact with it, what are the kinds of things that you get as responses?
Well, this is. I don't think that I can actually answer accurately, and give you an accurate answer to that. But I would say that the neurons would respond to stimulations in 2 different kind of ways. There's the reflexive, or the kind of immediate response. When you stimulate neurons, they respond. There's a barrage of activity.
Okay.
There's a barrage of activity that, subject to the way we patch the synthesizers, you hear this happening in various kind of ways, in various kind of aesthetics, and that would change between kind of patching. Um, but then the more interesting way of them responding is them changing their functional plasticity over time
Mm.
to control the synthesizers in a more interesting or less interesting way, hopefully a better way. Um, but, you know, and that kind of poses a little bit of a problem with Self, because our performances are only 1 hour, and 1 hour is not enough to kind of get neurons to learn, or to get neurons to
Right.
change.
Right.
So, I mean, the ultimate performance of Self would be over 48 to 72 hours, or 2 weeks even, where the musicians are playing with the cells every single day, all day long, or as much as we can, and the stimulations are kind of sent to the neurons over and over and over and over.
Right. Right.
So when we have these 1 hour performances, there is some sort of a symbolic gesture there, even though the neurons do respond and they do change sometimes. And if you listen through on the website to the concerts that are there, you can see that I think that those that the music or the concert is quite interesting. I mean, the neurons are doing things there,
Yeah.
and sometimes there's things that they do, and we kind of look at each other and say, "Oh my God, where did this?" So I can't really tell you that there is data that we produced that indicates that this is happening.
Yeah.
What we have is the concerts and really interesting observations. And I mean, we are not scientists. We're not there to produce data.
Right. Right.
The project, as I kind of said, is looking at the future, is looking at suggestion, and trying to critique, or trying to suggest those contestable futures, and trying to critique those technologies as much as we can through art.
Sure. Now, I'm really anxious to get into your background because that's one of the things I like talking about. But before we do that, there's one other question I have, because it actually is referred to a little bit in one of your previous works that you describe on your site. And for listeners, I'll have in the show notes, I'll have links to his website so you can dig into this a little more. But one thing I was curious about is to what extent there's any sort of like memory or kind of like embedded concept of a memory in these cellular cell-based techniques. And the reason I ask is you actually, the description of an earlier work, which is, I think, I remember it called Snowflakes. I think that there was actually a different name for it. But Snowflake, it was, you actually attempted to embed like a specific shape in the cellular network, and then you did a cryogenic freeze, and unfortunately the mechanism broke and it was lost, which actually, just in reading it, I actually had an emotional response to, which I guess is part of the artistic purpose, right? Is to get an emotional response to this as an idea, because you are toying with what is life, what is just technology, what is our manipulation of portions of life, right?
Yeah.
But that made me think about this idea of patterning and the concepts, what are the different concepts that make up memory? And I was wondering if in this work you talk about using this plasticity to sort of like change the way things that people, or that the system might respond to the people it's interacting with. Have you been able to experiment with it in long enough periods so that you could see where it would set itself up to maybe be very responsive to a certain player, or be very responsive to a certain kind of stimulation in a almost repeatable way?
Yeah, well, that's the $1 million question.
I see.
Uh,
Okay.
but no, we haven't. And as I said, I mean, it's an artistic project and not a scientific one, even though we're very interested in what exactly the neurons are doing.
Right.
Unfortunately, SymbioticA is very hard and expensive to install,
Mm-hmm.
and we don't have it set up at all times. So we haven't done kind of long-term experiments with SymbioticA.
I see.
But I'm trying to work with a few of my colleagues, scientists and artists, to put in a grant to kind of do it scientifically. And doing it scientifically is doing it in a much more controlled environment, where you can actually do things repeatedly without, because when we're taking SymbioticA on the road and we're installing it in galleries, there's noise and there's so
Right.
many that are kind of changing. You really need to do it, and you need to add a computer to kind of record the numbers and to kind of analyze them later on properly.
Right.
So we're not doing it.
I see.
But I think that the question that is in the heart, or the core of SymbioticA, is whether different rhythms and different ensembles would affect self in a different way,
Sure.
or functional plasticity in a different way. And I think that it's really interesting. I think, and a few neuroscientists really um, experienced and well-renowned kind of neuroscientists that I've been talking to about this project, they're all very interested in the idea of entraining of
Right.
neural networks to
Yeah.
see if something would emerge. And I think that it's more about the rhythmic behavior of the neurons and the kind of the rhythmic stimulations, or the rhythm of the stimulations that is being sent, or frequencies to kind of to the neurons.
Sure.
So I think that there's something there, and I think that it would be interesting to study. But Self did not collect any data. As I said, it's more about observations
Right.
that we
Right.
have through the music.
Fascinating. The combination of things that you do for your work is a pretty bizarre combination. I mean, from wanting to be David Bowie to harvesting your own cells seems like quite an interesting journey. How did you get to get drawn into this as a form of art? How did you choose to be an artist when it's clear that you also have this fascination with science? What is your route that you took to get where you are?
It's actually a very it's interesting because I mean nothing in my first 20 years of first 25 years could indicate this path that I chose later on. I grew up in Israel. I did grow up in a family that had quite appreciation to art. My mom has a quite an amazing collection of art in the house, and we would always go to exhibitions and to openings, but I never kind of thought that I would be a practicing artist. I studied law when I was in Israel, and then I didn't really like it. So after I graduated, I started in the mid 90s to work in as a programmer and technologist in the first kind of internet companies in Israel. But what really made the change was leaving Israel and going for a visit to visit my one of my best friends then, Oron Katz in Perth. And Oron, I met him in Israel, and in Perth he studied in Curtin University and he did his honors. And one, I can't really remember exactly what he did there, but he was the first one that I know that started to kind of look at the idea of using tissue culture as an artistic medium. And he got a couple of grants in the mid 90s. And when I came to visit him, he and his partner, Yonat Durst, just started their project, Tissue Culture and Art Project. And I was fascinated by their work. And, I just came to visit them for 6 months, but I never left. So I started to work with them, and I started to, because they were very lucky in the sense that they approached this scientist. I don't want to really talk too much about what happened to them, because I don't know if I can say it very accurately. But they approached a scientist in UWA, in the university where we work in, and she was very encouraging and accommodating, and they started to work in a lab. And when I arrived to Perth and I lived with them a little bit, the work that they did just blew my mind, and I started to go with them to the lab, and this is where it all opened. Not long after, I managed to get a job in the School of Anatomy and Human Biology, managing a microscopy lab, and this is where I realized that that's what I want to do. I mean, I started to kind of engage with science and started to engage, but in a more kind of artistic way. I worked for the first 3 years. I worked with Oren and Yonat on their own kind of projects, and I learned quite a lot from both of them. And then I started to develop my own work. So it was this move to Perth, and it was the interaction with Oron, mainly Oron, and also Yonat, that kind of did this shift in the way I kind of thought about my future and about what I want to do.
I see. That's really interesting. Now, one of the things I notice in looking at the different descriptions of your work is that a big part of what you do is sort of like a masterful gathering of a team, right? You talked about it in terms of cellf, but every one of your bigger projects seems to have this orientation of bringing together kind of like the extreme scientist and kind of extreme artists or musicians or whatever, and helping each to bring their own kind of voice or concept into the project. Um, what is the part of that makes up your voice? Is it this being the glue of collaboration, or are you working with them to help expand your voice into their area? Do you know what I'm saying? Do you find
Yeah.
yourself playing coordinator, or do you find yourself doing collaborations with everybody? How does that really work?
I understand. I understand exactly what you're asking, and thank you for asking that because I failed to talk about it before. Collaboration is a very important part of my work, and one of the reasons that I collaborate is that the projects that I usually aim to develop are quite complex.
Mm-hmm.
They're quite big, and there's just so much that I can do or know how to do. So usually those teams that I kind of build or the collaborations that I kind of form are mainly due to the fact that I don't know why, but I always think big. I always want to kind of create those projects that are quite complex. I mean, I'm basically shooting myself in the foot. I mean, the development of those projects are quite complicated, and usually takes me 4 years or 3 years. And when I started to work on self, I was very cautious about that. It's my self-portrait, and I decided that I'm just going to do it myself.
Right.
But then the things that I can do are quite limiting. I wanted to do way more than that. I wanted the project to go to different directions. And I understood that unless I collaborate, I would never produce the project that I want to produce.
Sure.
So really the reason that I work in those collaborative projects is this idea of interdisciplinary research, the idea that we can't do everything. We need to work with other people in order to kind of create complex projects.
Mm-hmm.
And then I also really enjoy it, because I think that the collaborative vibe or the people that I work with, especially in Cell, for instance, they made the project way better than I could ever dream of. The ideas started to flow from Darren, from Andrew, from Nathan Thompson, I'm working very close with on a new project. Well, we collaborate on a new project of that we kind of thought about together. The scientists, I mean, it was quite an amazing process of developing this project itself, because we were a big team, and there was no real kind of problems with ego or who is doing what. We all fell into our kind of beat that we know how to, and we worked very well together to produce a project that I could never dream of doing myself.
Right. When we take a look at some of these collaborators, I think actually a lot of my listeners will know Andrew Fitch, who is the person behind nonlinear circuits, right? His work with creating sort of chaotic electronic music tooling is pretty well known. But, you're working with other people like scientists in biological research. How do you even get people to have a common vocabulary? What kind of glossary do you have to build so that people can talk to one another?
Well, that's the ethos in Symbiotica. We work with scientists and we work with artists.
Ah.
And I'm an artist that's been working in scientific labs for 20 years now.
Right.
Nathan Thompson is a resident in Symbiotica currently as well. As I said, we're developing a project together. Andrew, he lives in Perth. He started to show up in Symbiotica as well. We had quite a lot of meetings. I mean, this is what's so special about Symbiotica, that the language is becoming the scientific language from one side, but the artistic vocabulary from the other side are starting to become very familiar.
Right.
Dr. Stuart Hodgetts, for instance, the person that one of the scientists that we're working with, he's been collaborating on dozens and dozens of artistic projects. When you hear him talk, he may sound like an artist sometimes. He claims that I sound like a scientist. We always have this kind of conversation. Douglas Buckham, another one of SymbioticA collaborators, he was a PhD student in the Potter Lab, and he was the person that helped me develop MiArt, this first project that I was talking about.
Mm-hmm.
And he was such a great person to work with. And through the years, once he kind of graduated, he said, listen, I don't want to be sucked into one of those projects again. I'm going and developing my own career. And then every couple of years, I would kind of send him an email and saying, are you free to work with me now? Because he's a brilliant neuroengineer.
Okay.
And then I was really lucky that a couple of months before SymbioticA, he sent me an email and said, okay, I do have the time now and the capacity. Let's do it. And, straight away he joined the project. So, he's been working on artistic projects for 10 years as well.
Right.
But it is this kind of space in SymbioticA that allows this kind of interaction between those, people talking about fertilization of those cultures, the science and the art. I'm more talking about the contamination. And that's where it becomes fun.
Right. Man, I'm looking at the clock and already our time is done. And I've got questions ranging from how it is that, you're able to reprogram. I mean, you talked about this idea of reprogramming cells into stem cells. It makes my head want to blow up. I don't even understand exactly what that means, but it sounds amazing. I wish I could explore more. I mean, I am going to ask you this. What is it about Perth that's making, like I said, my original introduction to you came from somebody that said, hey, there's some stuff happening here in Perth you might want to take a look at. And I started looking into it, and, with the Seattle Lombard modular systems coming out of there, the work that Nonlinear Circuits is doing, the stuff with coming out of Symbiotica, and your work in particular, what is it about Perth that is generating this kind of stuff?
Well, I don't know. It's really easy to work here. The lifestyle is quite high, and when I moved here, it was actually really cheap to live here. I mean, not anymore. But as far as biological arts, I think and it's a very easy place to live where people have time to kind of do their own thing. It's Oren, the director of Symbiotica, Oren Katz, always kind of says that he doesn't think that Symbiotica could happen anywhere else. It's only in Perth where it was this hotshot scientist that Oren approached, and rather than kind of looking and saying, "Hey, are you a scientist? I don't have a lot of time right now. I can't teach you anything." He said, "Well, this is my lab,
Right.
and you're welcome to come here and work whenever you want." And this is how the seed for Symbiotica kind of start for Oren. So I don't know. I think that the isolation may be one of those reasons.
Right. I was wondering about that, if the isolation doesn't offer the opportunity to sort of like develop your own cultural norm.
Yeah, I think so. I strongly believe in that.
Well, Guy, what a fascinating talk. I hope that the listeners will go onto your site and take a look at the work. I mean, it is pretty shocking and amazing, and it really does what you set out to do, which is make you start thinking about what is life, what is thought, what is consciousness, what is reaction, what are all of these things. It really does push that envelope of questioning. And I have to thank you for that, because I have to tell you that it caused me to ask myself questions I'd long ignored. So, fabulous work.
Thank you very much.
So with that, I'm afraid I'm going to have to we're going to have to stop for now, but I hope that we'll get a chance to talk again soon and further discuss some of this stuff because it is amazing. Before I do let you go, though, tell me a little bit. You said that it takes you years to develop these projects. Are you working? Are you still kind of like trying to finish up development on Self, or are you starting to work on a next project?
So no, Self is done. We're not further developing it, and
Okay,
we are performing with Self now. So
I see.
we're having there's two performances in Today's Art festival in The Hague in September, and then we're going with it to Helsinki for a couple of performances with by Art Society, and we're kind of fully booked until 2021, mainly in Europe. And while we're performing with Self, Nathan Thompson and I are collaborating to develop a new project called Bricolage. And really, in a one-liner is what we're trying to do is we're trying to, this time, this project doesn't involve neurons. First time after such a long time for me. We are trying to grow cardiomyocytes, so basically heart muscle cells, on various kind of scaffolds, miniature scaffolds that we're developing to produce artistic, autonomous biological robots that are powered by the movement of the cardiomyocytes, the heart muscle cells, over the structures. And the idea there is that those sculptures or those structures or biological robots are going to self-assemble through the exhibition. So we're going to have many of them that are going to self-assemble to one big one. But the movement of those, or the behavior, the autonomous behavior of those biological robots are going to be defined in the the behavior is going to be determined by the movement of the heart muscle cells on the structure. Because when you grow, this is what's interesting about heart muscle cells, when you grow them in tissue culture outside of the body, as opposed to other type of cells, they contract, they just kind of, they beat, they twitch
Right.
continuously. So we're trying to use those inherent properties of those cells and grow them in masses over really small structures to create those robots. So this project is less about data and the kind of manifestation or sonification of the data through movement or sound, but more about the visceral behavior of this biological material.
Very interesting. Very interesting. Well, look forward to seeing that in action as well. With that, I want to thank you. I will let you have the rest of your day, but I want to thank you for taking the time out of your schedule to have this chat. It was really fascinating.
Thank you for having me.
All right. And with that, we'll say goodbye. Bye now.
See ya. Bye-bye.
Thanks so much, everyone, for listening. I hope you really enjoyed this. If you have a chance, check out the cellf. He calls it self. I call it cellf project. It's crazy. The address is in the show notes. Otherwise, check in next week. We're going to have more great stuff coming around the pipe. And in the meantime, have a good summer. Catch you next time around. Bye.